Yeah, we're going to talk about lomas and Texas tortoises today. I want to first of all acknowledge all my co-authors because there are a lot of people worked on this project, most especially Juan Eliseche, whose master's thesis was uh, his master's thesis was the basis of this project, or this project was the basis for his thesis anyway. And he worked really hard collecting a lot of data out there, so we're really grateful for that. First of all, I just want to start with the basics because I know when I moved to South Texas, I didn't know what a loma was. So, lomas are elevated areas. They're very similar, mentally, maybe similar to sand dunes, but they're composed of more clayey soils. They could be any shape, really, but they're usually round, elliptical, or crescent shaped. They most likely formed millennia ago. Unlike sand dunes, they don't migrate. They just stay in one place. And they are host to a combination of Tamaulipan thorn scrub and grassland vegetation. Lomas can be found in North America, northern and western Africa, and southern Australia. The ones that we have in North America um, are still active. So in North America, they are found right along the coast and on either side of the Rio Grande. So in northern Mexico, in southern Texas, and in southern Texas, they're just in these four counties: Kleberg, Kenedy, Willacy, and Cameron. In other parts of the world, they aren't necessarily found in saline tidal flats, but the ones here in South Texas are. And how they form in these tidal flats is that there will be seasonal flooding, and eventually that will dry out and it becomes really cracked, dried mud. And as those cracks, continue to dry out, little pieces of clay um, get airborne because they are so light, and they'll get caught eventually on vegetation and they'll form a loma. And that just keeps happening over and over and over again. So these are still active, maybe growing, maybe shrinking. They're generally classified as one of three different kinds. The first kind of loma is evergreen loma. And these are classifications largely uh, of Texas Parks and Wildlife. And the evergreen loma has a really dense layer of shrubs, and there's a wide variety, but there's a few mentioned there on your screen. Years and years ago, after a meeting I saw on a news conference, the mayor of Corpus Christi said, and I'd never heard this before, he said everybody was jam up and jelly tight in there. And I'd like to think that I'm not a hillbilly enough to know what jam up and jelly tight means, but if I do kind of have an idea what it means, this is what it means. These brush species are just so tight in there. The plants are so tight. You can see one trying to crawl out of them there. You cannot walk through them. Every space is taken up for a branch, but you can imagine if you were a tortoise on the ground, this might look really attractive. The second kind of loma is a deciduous loma, and the species are similar to that evergreen loma, a lot of woody species, but the density is reduced. So maybe you can walk through them in most places. And then lastly, there are grassland lomas that look just like what you would imagine, mostly grasses, but there what might be a few scattered woody plants in there. So this project was funded by Texas Parks and Wildlife Department, and our objectives and hypothesis are these main three. First of all, we wanted to determine the soil texture and pH of lomas and their surrounding areas. And we hypothesized that the soil texture and pH would be different on the lomas compared to the areas surrounding them. Then also we wanted to quantify and describe the plant species composition, the cover, the density on lomas, and we hypothesized that it would vary among lomas and also among sites. Last, we wanted to determine the density of Texas tortoises, and we were thinking that they would be um, more dense, that there would be higher numbers of Texas tortoises on lomas per hectare than what is reported in the mainland, which is basically 0.26 tortoises per hectare, or about one tortoise per every 4 hectares, one tortoise for every 10 acres. Here are our 3 study sites. There's El Saus, which is a privately owned ranch owned by East Foundation, and it's right there near Raymondville, right on the coast. The second site in the middle of your screen there is Next Decade Rio Grande LNG. LNG stands for liquid natural gas. And then Laguna Atascosa National Wildlife Refuge is our third site. Originally, we planned to put 5 lomas, the first 5 lomas, at El Saus. And so we did, but we had to come back to El Saus in the end, put the last 4 on there because a neighboring landowner that we were trying to work with, they were being really cooperative with us, but we couldn't find a time between seasonal flooding. We couldn't get out to the coast on them, or they had hunters. So we just came back to El Saus for those last 4. The Next Decade Rio Grande is just one Loma, and it is the largest Loma, maybe one of the largest Lomas down there, almost 100 acres. And then 4 Lomas on the refuge that range up to like 7, almost 8 hectares there. So let's start with the soils. I need to orient you a little bit to what we did so that you can understand the graphs and such. The first thing that we did is to outline the perimeter of the Loma. So the Loma you see there in blue, we outlined that, and then we have 2, so we marked the Loma, and then we have 2 transects. Transect 1 goes from the southeast corner on your bottom right up to the northwest corner, and transect 2 goes from southwest up to the northeast, and they cross at the midpoint of the Loma. Now, later on, we're going to talk about inside southeast, that's inside the Loma on that first transect in the southeast corner, inside southwest, inside northeast, inside northwest, and then outside the Lomas. And on those transects outside the Lomas, we started at the edge. We put 10 points going away every 10 meters. So on the outside of the Loma, they range out to 100 meters away. We collected soil samples at those 10-meter intervals. Those soil samples we brought back to the lab, and we did the stuff there in the lab. Okay, so I'm going to keep showing you that diagram of the Lomas and the transects so that you can remember what I'm talking about. And let me orient you to this graph too. It's a little bit, there's a lot going on here. So center is the center of the Loma. INE is that inside northeast. So all those points along that transect, inside and northeast of the center, and then inside northwest, inside southeast, and inside southwest. And you can see that the center point, elevation wise, is a little bit higher than the inside northeast, inside northwest, inside southeast, but it doesn't vary statistically from the inside southwest. What's going on here on these graphs, the box and whisker thing, the dark bar in the middle of the box and whisker is the median. The box itself represents the first and third quartile. The whiskers are the range. Those couple of spots up above, circles up above, are outliers. And then because we were working with the median because the mean can be pulled, up or down by an outlier. But we wanted to show you the mean, and the mean is what's going on there in those red dots. The red dot is the mean for each one, the mean elevation, and the whiskers there are the standard errors of the mean. So over here in the lower right corner of your screen, the maximum elevation ranges, ranged from about 4.5 to about 7.5 meters. The minimum, we're right there at sea level. So from less than a meter to almost a meter and a half. And the averages were about 2.5 to the lower three. Now, we didn't do statistics on the elevation outside the Lomas, but I wanted to show you this graph. If you look at the group on the far left that has the SE under it, the dark bar is the 0 meters. So that is right at the southeast edge of the Loma. And then as the colors get lighter, they go out to the 100-meter point. And so for each one, you can see the vast majority of the difference in elevation happens right there in those first 10 meters. You step off that Loma and you step down, and there's a big difference in those first 10 meters, and then it's much more gradual after that. As far as pH is concerned, let's start with inside the Lomas. Inside the Lomas, we have those box and whisker graphs again, and there's a lot of variability. You can see by the whiskers, there's a lot of variability there, but there's no difference in the pH. And now I'm saying that there's a lot of variability, but if you look at the scale over on the left, the scale over on the left starts at 7.0, finishes at 8. So those medians, those black bars, the medians, they only range from like 7.7 to 7.8. So there is variation, but the medians are pretty tight, and no difference inside the Lomas. Now, if we look at pH and we compare pH inside the Loma to outside the Loma, now we have the southeast, I'm sorry, at the top, we have the southwest 50 meters from the edge of the Loma. And then we have right at the edge of the Loma, and then the center of the Loma, and then continuing on that transect, the northeast at the edge of the Loma, and 50 meters from the edge at the top of the graph. There's nothing going on there. If you can see our p-values there, 0.3 for both transects. The pH doesn't vary off the Loma as opposed to on the Loma. That's the bottom line there. Again, just on the alkaline side, just above 7. Now let's look at soil texture, and I want you to look at these two graphs first with the red, green, and blue bars. Both of these bars represent one of the transects, and the middle bar there is the center of the Loma. Like the last slide, the first bar is going to be 50 meters out. The second bar is the edge. The fourth bar is the other edge, and the fifth bar is 50 meters out too. Oh, I hope you can see my pointer. I just realized I could do that. So anyway, absent-minded professor here. So in the center, these are both the same point, and they are both clay. Now look at our p-values down here for both transects, 0.4 and 0.3. There's nothing going on statistically. Statistics say, hey, all of those guys, they have similar textures, similar percents, sand, clay, and silt. But I wanted to put this soil triangle up in the left corner for you, because if you're like me, maybe you haven't paid a lot of attention to that in a while. And even though the statistics tell us they aren't different, look, they do fall out differently on the soil texture. And what's interesting to me is that if you look at the first bar, on the south side, southeast 50 meters apart, or southwest 50 meters apart, or 50 meters away, southeast and southwest were both silty clays. If you go right to the southern edge, the southeast edge, the southwest edge, those are both sandy clays. So they're a little siltier farther out. They're sandier as you get close at the edge of the Loma. And then the north edge of the Loma, both sides on the northwest and the northeast, these are sandy clay loams. And then 50 meters out on the northwest side, there's sandy clays. And we did make sure that we had at least 200 meters between each Loma so that even the last point was not being affected by its proximity to the next Loma. So it's interesting. Statistically, they're not different, but they do fall out differently on the soil classification triangle. So first, our data don't support our hypothesis that pH and soil texture are statistically different on the lomas. However, those classifications, those texture classifications at different points on the Loma or off the Loma, they did vary, and that does, as you all know, make a difference to vegetation. We also compared these to the USDA ecological sites. Like if you look at your web soil survey, they're going to call what we're calling Lomas, they're going to call that a coastal ridge site. And they say the pH ranges from about 7.4 to 9 on those. Our pHs were 7.5 to 7.7. So we're right in that range and a little bit tighter. And then they call the area surrounding the coastal ridge sites, they mostly call that wind tidal flats or those lagoons in between. And they list pH of 6.6 to 9. And again, we're right in there, just a little bit tighter. Let's talk about vegetation. And we did two different things for herbaceous vegetation and for woody vegetation. So I'm going to start with herbaceous vegetation. For herbaceous vegetation, at first it was just a very, very much a let's look and see what we have. And so we approached each Loma and looked at the herbaceous plants out there, and we tried to identify different communities. Like we would say, okay, over here we've got a Gulf cordgrass community. Over here we have a shoregrass community. Over here we've got seacoast bluestem. And so we would identify these communities and try to go to this pretty much the center of the community, and we would lay out a 5 by 5 meter plot square. And then within that square, we put this 1/4 meter frame at each corner and one smack in the center. And in those 1/4 meter frames, we recorded percent bare ground. We counted individuals of every species that we found in there, and we estimated the percent cover for each species. For the woody vegetation, we had to do something a little bit different. So here's our Loma again in the blue. And what we did first was we marked each clump of woody plants. Now, there might be some individuals also, and they got counted as well, or not counted, but their species were recorded. If it was a small clump, you might be able to see everything in it. And that was cool. But for some of these bigger clumps that are too thick to even really crawl through very effectively, what we decided to do was we would walk around the clump and say, okay, I can see in about 3 meters here. I can only see in about 2.5 meters here. I can see in 5 meters here. And we would record and sort of get an average of how far we could see in on every clump. Every clump was numbered, and every clump got a width that we could see in to go along with our perimeter so that we could get an area. And then what we did was we recorded all the woody species that we saw. We weren't counting them. We were just recording species. So it was frequency. And we also recorded the average height of each clump of brush. We did an ocular estimation of percent canopy cover. Now, when we got started on this project and we were trying to work out how are we going to do things, sometimes we would see Texas tortoises, and we would often see their feces. And once you get the feces in your field of vision, it was like, whoa, it's everywhere. So we often noticed that we also saw Texan goatbush at the same time. We would even see little pallets where you could tell that a tortoise had made a little pallet. Even if you didn't see the tortoise in there at the time, there was a little pallet of a tortoise underneath a Texan goatbush. And Juan did see some individual tortoises during the right season of the year where their mouths would just be red with the Texan goatbush berries. So we saw, we sort of thought, you know what, this might be important to them. So we decided that we were going to count every single individual Texan goat bush that we could find. And so we did that in every clump where we could see the whole thing. And we did that in those fixed width. We were counting those guys instead of just recording that it was there. All right. So there is a lot of there were a lot of things that I could talk about. And I thought, you know what, let's just do a descriptive, what grows on these things. So this is the herbaceous vegetation, sort of the common species, the unique species, the non-native species. At El Sal's, one of the most common communities was shoregrass, and it was also really common at the Rio Grande LNG Loma. Also common at El Sal's was Seoxi and leafless cressa. At El Sal's, there was red grama, and we didn't find it anywhere else. And also at El Sal's, we did record some King Ranch bluestem and some buffalo grass. At the refuge, we most commonly ran into silver bluestem or pink thoroughwort or plains bristlegrass. And they did have some scarlet pimpernel and Arizona cotton top that we didn't find at the other two sites. As far as non-natives are concerned, there was Kleberg bluestem, guinea grass, and buffalo grass. And the refuge probably had the most of the non-natives, and the majority of the non-native grasses there was the Kleberg bluestem. At the Rio Grande LNG, there was leatherleaf in addition to that shore grass. They had silky evolvulus, which we didn't see anywhere else, and they did have some guinea grass and buffalo grass. For woody vegetation at El Sal's, well, actually, so at all three sites, there was coyotillo, loaf bush, and tornillo as very common. And El Sal's in the Rio Grande LNG also had Texan goat bush, very common, whereas the refuge had Spanish dagger as a very common species. It was found at the others, just not as common. Unique to them were Texas ebony was unique at El Sal's. The refuge had some things that didn't show up other places, and you'll see this later. They had some bird pepper, ratama, snake eyes, desert yaupon, and then the fiddlewood we found at the Rio Grande LNG site. We did not find any non-native woody plants. So I want to show you some ordinations that we did. This is very much a multivariate community analysis out there. So I didn't want to, like look at p-values and things like that, but we want to look at how these communities are similar and how they are different. And so over on the left side of the screen, you remember we identified vegetation communities at each site. So we have this ordination here of the communities, and you can see most of them are bunched up here together because these three really different ones are spaced sort of far away from them. And this one is the Kleberg bluestem community that was so common at the refuge. And then the Gulf cordgrass communities at the other two sites are a little bit different than the other communities. And you can see the species over here that are pulling these sites away, the Kleberg bluestem. And in the Gulf cordgrass community, there's also soldier weed and partridge pea, and that's pulling them away. So what can we do about this here? We thought, if we take out these outliers here, we can get a better look at what's going on inside that group of the most common ones. So this is the same thing with those outliers gone. And now, because there are fewer, we can put a little anchor to them to help us see them on the second axis. And you can see, okay, so green is the next decade Rio Grande LNG site, and red is El Sal's, and they pretty much bunch up here together. But Laguna Atascosa has some communities that are off by themselves, and we see the same thing here. Okay, they're pretty much bunched up, and there are going to be some individuals off by themselves there. But what we see here, what's most important to think about here really is that Laguna Atascosa is really kind of different from the other two. Now, if we look at the woodies, for woody vegetation, we've just done the ordination by Loma. So we've got 15 sites in here. The green now are El Sal's, the blue is next decade, and now the red are um, the wildlife refuge. And again, the wildlife refuge with the woodies also is making itself a little bit different than the others, similar to what it did with the herbaceous species. Snake eyes is this species here, the PHSP, and it was only found on Loma 11. So hopefully you can see my pointer there. CAANG is bird pepper. It was only found on Loma 8. Rotama is the PAAC, only found on Loma 10. Desert Yopon, only found on Loma 9. And so when we do these ordinations, a lot of times those unique species that aren't found in other places are what pull these communities apart. So overall, very diverse communities. And these data do support our hypothesis that vegetation composition would vary among the sites, and it would also vary among the lomas. There are some environmental factors, also historic, management factors that might be causing differences in vegetation. El Sal's is right on the coast, and it's also privately managed. It is a working ranch, and so every once in a while you would see a cow on a loma, and more often than that you might see cow patties. So that's a little bit different probably than the other two. At Laguna Atascosa, because that is under public management, maybe the visitors are the reason why the non-native grasses are a bigger problem there than at the other two sites. And the next decade site is close to the Rio Grande River. I kind of glossed over it, but it is right on the Brownsville, the shipping lane. So it is right on the river. Now let's talk about the Texas tortoise. For tortoises on each Loma, we did 50-meter belt transects, and 3 people that were 10 meters wide. 3 people would walk these transects, and we walked them, during their active months, during the active times of the day, one on the center line and one on either side. And the ones on either side would sort of move back and forth, looking under vegetation, looking for tortoises, and the one in the middle tried to hold that center line while also looking for tortoises. All tortoises were marked, as far as their location is concerned. Now, that's what we did on 14 of the Lomas. On that Rio Grande LNG Loma, there was a 100% census of all tortoises done, and I will talk to you about why that was done in a second. But let's just look at what we found as far as Texas tortoise density is concerned. At El Sal's, 5 of the 9 Lomas had tortoises. We found 15. At the refuge, 2 of the 5 had them. We found 5. On that Big Daddy Loma, there were 148 tortoises at that Loma. And so that works out to, when we look at our area surveyed and our number of tortoises, that's about 3.9 tortoises per hectare. And as far as the goat bush is concerned, we did find that the presence of tortoises is correlated with the presence of goat bush. In other words, we were more likely to find tortoises where we also found goat bush. In fact, 83% of the Lomas that had goat bush, we also found tortoises on them, but only 25% of Lomas that didn't have any goat bush had tortoises. So that seems like an important component. Now, the density, there was not a relationship. We did not find a relationship between density of goat bush and density of tortoises. In other words, where we found more goat bush didn't necessarily mean that we were going to find more tortoises. And this part, if you're interested in more information on this, This part has already been published as we combined this with information from another study. So that's Moeller et al. In 2025, habitat preferences of Texas tortoises, yada yada, in herpetology notes. So the data about on the tortoises do also support our hypothesis that tortoise density would be greater on Lomas than what's been reported for the mainland. And we can see that Lomas provide prime habitat for Texas tortoises. We recommend that conserving Lomas for the Texas tortoises should be a priority, particularly Lomas with Texan goat bush. And Lomas should be monitored for invasion of the non-native grasses so that we can take care of them quickly. You can see, what happens with these non-native grasses. They're just so thick. And if you look at the picture on the bottom, imagine being a tortoise trying to make a living in that. It just doesn't work very well. All right, so this always comes up, and I have permission from my good friend and colleague, Dr. Scott Hinkey, also at Caesar Kleberg Wildlife Research Institute, to share with you a little bit about this, because this was actually part of his study. That next decade, LNG Loma we used for this study, he also used for one of his studies. That Loma, it was cleared, it was leveled, and it was paved over for development. They are an LNG company, okay? In their defense, they did give Dr. Hinkey a big check to study for a translocation study of all the tortoises. So every tortoise, that was searched really well, and even as they were working, if they found ones that Kristen missed with her thesis work, they would gather them and give them to Kristen. So Scott and Kristen set up 3 soft release pens. They were 5 hectares each. And those soft release pens, one of them held tortoises for 4 months. They were all started on the same day. One of them held tortoises for 4 months, one for 8 months, and one for 12 months. And so when that time had passed, they just took the walls up, and tortoises were free to come and go as they wanted to. And of course, so they wanted to determine the survival of those tortoises, and is there any difference in movement patterns if you hold them longer as opposed to a shorter amount of time, and are there any sex differences? And I just want to show you again, I do have permission from Scott to share this, and everybody always asks about that Loma, so I figured I'm just going to go ahead and show you. These are some of the tortoises that were kept there. The black rectangle with a little chunk taken out of it, that's our South Pasture facility on the south side of Kingsville. This female on the left, she was held in the 12-month pen, and she did exhibit what might be considered potentially homing activity. She traveled south about 5 kilometers within a year. Over on the right, this is another female, both adults, held in the 8-month pen. She traveled west. She hung out here where the green dots are for a little while, and then in the summer, she traveled northeast again and set up a new home range. On the left, this is a male from the 12-month pen. This male took off heading west, made a great big old loop-de-loop, came all the way back, went even farther south, came even farther east, and has ended up back where at the recipient site. And then this female was just from a 4-month pen. And when her pen was open, she made this little excursion, but has spent the rest of the time here and is back at the recipient site too. So what Scott has found in his study, and I don't want to scoop it too much because he is working on the manuscript. He's literally waiting on edits from me. So this will be out soon. Anyway, he found no differences in mortality among the pens. All of the survivorship from 4-month, 8-month, 12-month, it all ranged from about 70 to about 95% survivorship. They did see 4 instances of mating in the pens, two in both the 8 and the 12-month pen. They never saw any hatchlings, though, and they found no differences between sexes or among release pens. These tortoises moved in all directions and distances in very individualized patterns, regardless of treatment. So, future research recommendation on the lomas. I think one really important study would be a human dimensions study of what is the best way to protect these lomas from future development. Is it going to be through economic incentives? Does it have to be legislative? How can we protect the lomas that are remaining when everybody from SpaceX to LNG companies to private developers wants a piece of them because they are on the coast? Also, what is the most effective means for removing the non-native grasses without harming the lomas, because they are special sites? Maybe we need to do something a little bit more carefully there. And then I think what would make a really cool study would be the island biogeography of the tortoises. I actually wrote a grant to the Texas comptroller for this, but it didn't get funded. But Dr. Hinkie and I wanted to look at this island biogeography idea. How much movement and breeding is there among the lomas? We know that tortoises come off the Loma every once in a while, because we actually did see 2 tortoises out in the middle of the sand, just walking across the sand. One of them actually did it at like 1 o'clock in the afternoon on a summer day, and it was pretty hot. And we thought, wow, what's that guy doing out there? But also, if you're on these lomas, you can kind of look around and think, if you were a tortoise, maybe you could maybe live your entire life here on this one Loma, possibly without ever having to leave. So how much are they leaving and visiting neighboring Lomas or visiting the mainland? We wanted to track their movement and collect genetic information to be able to compare among and between. So hopefully somebody will carry that torch forward. I want to especially thank the Texas Parks and Wildlife Department for the funding and the landowners who allowed us to work on their property. Also, Juan did receive some tuition assistance from South Texas Quail Coalition and the Cesar Kleberg Foundation. We had some great technicians and fellow graduate students from my lab and from Dr. Hinkie's lab. I think I have time for questions. I actually think I went a little bit faster than I expected to. I think that's always the way. So, yeah, I have time for questions.
Sandra, this is Jason. Can you hear me?
Yes.
Okay. How do we? So that'll be in the chat. Do you see the chat on your end? The lower right.
B.
Yeah. Let's see questions on this side of. Okay. Okay. Here's a question that came up. Okay. First question. Sorry, it took me a second. How do you suggest controlling guinea grass? Or any of those exotic grasses that like to occupy low
yeah. Thanks for starting out with softball there. I appreciate it. So, great question. Pardon?
Go ahead, sorry.
I'll be real honest. I've worked with these questions for a long time, and I don't have anything super creative. But, obviously nothing mechanical out there, and I don't even know that mechanical would be a good idea anyway, but for buffalo grass. But I wouldn't be opposed to trying to burn them first, because maybe that's just the burner in me, but maybe burning first to get rid of all the dead thatch that's going to get in the way, and then treating with herbicide when all you've got are fresh green stems that can and leaves that can take up the herbicide. Burning by itself absolutely will not work. Mechanical is out. So it's going to have to be a combination, I think, of chemical with something else, or chemical maybe even by itself. And I would prioritize the ones that have not been inundated yet. Starting, if you can identify when there's just a spot coming in, get those spots sprayed and try to keep an eye on them, and then maybe work on some creative ideas where they have taken over. Yeah.
And let me kind of add kind of part B to that. Yeah, I mean, during your study, obviously you showed there were at these different study sites that you had sites that had were dominated by the red grama or silver blue stem, or at least some of our native grasses. So it sounds like identifying those lomas that have most of the native vegetation, but like you were just saying, have little quilt of the bases to try to treat those. Because in my experience, some of the Loma sites are just covered in the basic grasses. And I guess my question to you would be, kind of follow up would be, on those ones that really, really cover up in the bases, how would we even go about, um, once you treat it, how would you reoccupy, the herbaceous flora, aside, on these sites, as far as plant materials and so forth?
Yeah, that's a good question. So it might have, yes, great question. It might have to be because they are right there on the coast. I could see where they might be washed out if you had a hurricane when they didn't have any good vegetation on them, kind of thing. So, maybe partners, say like with the South Texas native or Texas native seeds, to
Yeah.
get them treated, get them, killed, and then try to get some natives established as quickly as possible. And
Okay.
yeah, it. I think it's going to be even more tricky than on the mainland because they are such special soil formations.
Okay. Sandra, we have several questions here now popping
Yeah,
up.
yeah.
Let's see. I suppose it's obvious, but there currently is no protection for Texas tortoise in the state? Question mark.
I apologize. They are state endangered. So,
Okay.
yeah, I should have said that. They are, I'm sorry, threatened. They
Yeah,
are
no,
state,
our
sorry.
department has state protection over it. Correct. Yeah.
Yeah, yeah.
Um, Vanessa, that was JC. Vanessa D. Will this presentation be provided after this meeting, including the slides? Yes, we'll post this in a few weeks. The video will go through our department, through our communications folks, um, and we'll post it in a few weeks. We have to look at the script and everything, and then we'll post it. Let's see. Based on your findings, Sandra, what would you prioritize in future restoration studies with Texas tortoise in mind? Soil condition, vegetation composition, or structural diversity?
Okay. I'm sorry. Let me see that
And I can
again.
repeat that.
Yeah.
So she says here, based on your findings, what would you prioritize in future restoration studies with Texas tortoise in mind? Soil condition, vegetation composition, or structural diversity?
Yeah, yeah. Hi, Kayla. Good question. Um, I really feel like it's vegetation composition for them. Now, vegetation composition uh depends on your soil, but I feel like it's that gosh, I think both composition, and now that I'm hearing myself say that, diversity of the structure, because I do feel like they really like that thick Tamaulipan thorn scrub where they can just get under there and very few other things can. So that thick structure, I think, is important.
Okay. Molly Smith says here, the mortality rate of approximately 69 to 94% seems like a pretty wide range. How was this determined?
So that's from Scott's study. um, the translocation study. But those animals had trackers on them, and when they got a mortality signal or when it was stayed in place for a long time, they went to find it, and that's how it was determined. And it was just so let me be more clear. It is a mortality rate and a suspected mortality rate, because now we're like, now that we're a year out and not all of them got receivers, many of them did, most of them did. I'm probably butchering this. Scott, if you're on, maybe you can put that in the chat. I don't know. But it was the suspected survival and the survival, but mostly comes from those ones that were tagged. So so
Um,
so some of them, there's some unsurety about some of them, I should say, because on private lands, I will say this: on private lands, like when Scott and Kristen saw that they were getting a signal of an animal that had been in one place for a long time, they would have to ask, hey, can we go in your backyard and see what's going on there? And some people said no, probably because it is a state threatened species, it is a protected species. Some people did say no. So there are some animals that they aren't sure what the outcome was. Sorry about that. I hope that's more clear.
that sounds good. Okay, another question from Mitch. Sorry, I missed the beginning. Were each of the three Lomas / sites only characteristics for one Loma per site?
Yeah, that's a good question. No,
Yeah.
we used 15 lomas, and we tried to go with 4 sites, but one of them we just couldn't work out the timing, so we ended up with 3 sites. The LNG site only had one, that big daddy Loma on it, and um, the other 2 sites had the other 14 between them.
And another question here. Curious to know what your impression is for vegetation differences, species composition, structure, etc., for Lomas near those they studied in general.
You know, because because we did do 15 and we sampled the dog out of those guys, we really did. I can't cover it in 45 minutes, and I wanted to talk about soils and I wanted to talk about tortoises, but we could have spent, like, I don't know, all day just about the vegetation and those those manuscripts. I'm still working on that, but I do feel like we got a pretty decent idea of the range of what you might find out there between the 15 different ones that we looked at. And I will just say that they're just incredibly diverse. They aren't just different from one to another, but the diversity when you're standing on a Loma, the diversity, especially in the thorn scrub, is just wild. And you really see that they're unique and they're special in and amongst themselves, but also, this one is unique from the one next to it. But I do think we kind of got a pretty good idea of the range between the 15 that we looked at.
Fantastic. Let's see here. Oh, hold on here. A bunch of questions popped in really quickly here. Screaming. Okay. I'm a fan of prescribed fire, but it seems that fire may be damaging to the evergreen Lomas. And maybe to add to that question is, Sandra, is it, I mean,
Mhm.
you have, I think that naturally these Lomas would have been more of that sort of mixed grass. And now with the invasives, it's creating a much larger, more dense structure. Does that make sense? So do you think that
Yeah, yeah.
prescribed fire
Yeah.
might be, yeah.
Great question. I'm glad you asked that, since I did put myself out there with the brown burn and brown, or brown and burn uh, combination. So let me say this. At El Sal's, I had a long-term burning study at El Sal's, that went from 2016 to 2024 or something like that, and we burned two large units of 500 acres to 1,300 acres. We burned two in the summer, we burned two in the winter, every year, all those years. And and we were mostly working in the grasslands right by the coast. So we're burning these plots right by the coast, like the coast might be my eastern fire line, and there's Lomas right there, okay, off the coast, or on the coast. And so when what I want to say is that I was at first a little bit worried about the woody uh, plants down there, but when they grow in a big clump like that, there's very little grass in them. And I can show you the coolest video of that they took at El Sal's, that they took from a drone. They flew a drone on one time and they videotaped our prescribed fire. Our fire just goes around those clumps of thorn scrub because there's no grass in them, and the fire is moving pretty quickly through the grass, and it takes a lot more heat for a lot longer residence time to get those. Like, maybe it could happen under wildfire conditions. I'm not saying that Tamaulipan thorn scrub can't burn. You did not hear me say that. It could probably burn under wildfire conditions, but under prescribed fire conditions, it just burns in the grass and it just goes right around it and comes right back together on the other side. And so they were skipping those thorn scrub things. So I want to say that under prescribed fire conditions, and that was a lot of experience that I got right there in that same habitat, same grasses, same Tamaulipan thorn scrub. We never got fire to go into that thorn scrub. Never.
Excellent. We have at least four more questions in a few minutes, so let's see if we can get through these together. What exactly does state in danger mean for species if a company can pave over habitat for 148 tortoises? That seems like
Yeah.
a huge number of tortoises.
Yeah, that's sort of a sob story there, that Loma. And um I I think maybe Jason and Jonah would have a better, surely there was some something that they had to do, and maybe it was the you know giving Dr. Hinkie the big check to do the translocation study. Maybe that's how they were able to do that. I do not know. I will say one more thing, and if you're not already in tears, I'm sorry, I might ruin your Wednesday afternoon. That LNG company, after they did that, they weren't able to get the permits. My understanding is they weren't able to go on with their construction, so that was all for nothing. So sorry.
Okay. Yeah, I'm crying
Yeah,
right now. Um,
yeah, I know, right? Just made
uh,
everybody cry in their coffee. Everybody's got to take the afternoon off now.
okay, three more questions here real quick. Hey, my boss that hired me at Parks and Wildlife, Dr. David Diamond of Missouri, says how many
Yeah.
lomas are there? I guess the total area.
Oh
Yeah,
my
how many
gosh.
lomas in total area? I guess you have that from your mapping work.
Uh, I do. I don't have it off the top of my head, but the next thing, we didn't talk about this, but the next thing, Ikai Hung at Stephen F. Austin State University, he took our information and then he was able to sort of map out where other lomas across the South Texas area might be found, and sort of mark these areas. I do have that information. I do not have it off the top of my head. I can email you that information if you want it. Sorry about that. I don't even want to make a guess. I apologize.
Oh, and David says here DPW does have a Loma map. Okay.
Yes.
Um, yeah, yeah, and so yeah, we can definitely with his data answer that. I'm happy to work with you to provide that. Those number of lomas and acreage to David.
Yeah, yeah.
Um, let's see. Oh, just a great shout out here. Uh, it says here, I'm sure. Okay, not need to reply. Cool. 15 lomas covers great differences. I'm sure. Thanks for all the hard work and so great, but I think it's kind of funny for you crawling around on your hands and knees and mapping
Mhm.
besides
Yeah,
photo methods. Yeah, it's a tough landscape to work in,
it is.
along with sea ticks